the triple antibody sandwich ELISA, which we know is used for HBV virus identification for the blood and all that, and that is the indirect ELISA. Now, in this module, we will discuss the double antibody sandwich ELISA and another technique to detect the antibody concentrations and antibody presence. Now, this is the most widely used immunochemical technique in the diagnosis. This, the double sandwich antibody technique, as a matter of fact, is most widely used and routinely used as immunochemical technique for the diagnosis and uses as a diagnostic test. If we see in this diagram that the ELISA plate, which is coated with the antibody, then the sample which contains the antigen is added, it is bound or it is not further, then antibody which is trapped for the antibody or antigen which is trapped to the antibody in there. Further, the antibody enzyme which is added over there and it reacts with them. Further, the color development is there, and we can determine that whether the antigens are present there or not. So this is the double antibody sandwich ELISA. You see, there is an antibody, antigen, and then there is an enzyme which is added to the plate which containing the color, and the color is determined, and from the color determination we can find out whether it is not there. Now, the advantage of this technique is that it is rapid, it is robust, and it is reliable. It is very reliable. Robust is that it can be performed more and more. It can give more results, and without any hitch, we can get significant, large results by using this technique. Now, the principle of this technique is basically the same as other ELISA techniques, as we have seen in the previous diagram, that the titer plate is there, the antibody coating, antigen addition, but only difference is that we are adding another enzyme which gives the coloration, and from the coloration we can determine this. So the antigen is immobilized to the solid phase by a primary antibody, which is a titer plate, then detected with a second antibody which has been labeled with a marker enzyme. As I said earlier, that marker enzyme would mean an enzyme which can indicate that whether this substrate is present or not. Then the antigen, it creates a bridge between the two antibodies. There are two antibodies, and it is in between those two antibodies that antigen is there. And then this antigen, it changes in color of chromogenic substance. That the antigen has the color of different color which is there, and the presence of enzyme, it causes a color change in the chromogenic color producing substance. So this is, in addition, there is a enzyme, a marker enzyme, and the change in the color of chromogenic substance that occurs, and by reading that color, we can check, we can determine that whether the antibody is present or not. Now, generally the marker enzymes which are used, they are horseradish peroxidases, HRP, or alkaline phosphatase, that is the AP. These are the two marker enzymes which are basically used there. In some system, the enzyme is replaced with a radioactivity label, and this format is known as immune radiometric assay, IRMA. So you see that during the diagnostic process, we use different marker. In the previous one, we used a marker enzyme, which gives a chromogenic reaction, that is the production of color. Now we need some detection. Another precise or very precisely determined thing is radioactivity. So what we can do is that we can use the enzyme, or we can replace the enzyme with a radioactivity label, and this format is also very precise and is known as IRMA, which stands for immune radiometric assay method, and it is very, very precise. Now, where we can use the double antibody sandwich ELISA method. Generally, it is used in horticulture and agriculture to ensure that the plant material, it is free of viruses. You see, the plants also contain viruses, some dangerous viruses, some viruses which cause disease in the plant, and DAS is used frequently for that. Now, there are, for example, many potato viruses, but potato leaf roll viruses, PLRV, in particular, it causes considerable problem and damage to the potato crop, and we can detect by this method. PLRV antibodies, they are coated on the wells of ELISA plate, and then the sap to be tested is added, as is done with the blood, and after incubation the plates are washed. PLRV antibody, they conjugate to the alkaline phosphatase, is added, and the plates are incubated, and after washing substrate is used to identify the positive wells. The system again requires presence of antigen, that is the PLRV antigen, for the sandwich of antibody to be built up. So you see, in this segment, we have discussed that how these methods, they are applicable not only to the human being, but for the plants as well, or wherever we have to determine the antibody-antigen reaction.